He'll chair professorship in the departments of biomedical engineering, orthopedics, pharmacology, and regenerative medicine, as well as mechanical and industrial engineering at the University of Illinois at Chicago. He was previously a professor at Case Western Reserve University in the departments of biomedical engineering, orthopedic surgery from 2005 to 2018. He received his bachelor's in biomedical engineering and in mechanical engineering and material science from Duke in 1994. He went to graduate school at the University of Michigan, where he received his master's in mechanical engineering in 1998 and master's in biomedical engineering also in 1998 and a PhD in biomedical engineering in 2002. Following his graduate work, he was the postdoctoral research fellow in the vascular biology program at Harvard Medical School. He authored more than 155 research papers and book chapters and has been recognized with the Tissue Engineering and Regenerative Medicine International Society Senior Scientist Award, Bio Valley Young Investigator Award from the Tissue Engineering Society, Ellison Medical Foundation New Scholar in Aging Award, and many other awards. He is also an elected fellow of the National Academy of Inventors, the American Institute for Medical and Biological Engineering, and Biomedical Engineering Society. He also has 35 issued or pending patents in the field of tissue engineering. The floor is yours, Professor.
Thank you very much, Mehmet. I really appreciate that introduction, and I'm grateful to you and Ali for inviting me to share some of our work today. I've enjoyed you guys' friendship and collaborative efforts for many years now, and so great to be reconnected today. Yeah, I'm looking forward to sharing some of our work. Please, everyone, feel free to interrupt if you'd like. Yeah, so I'll get started. So today I'm going to talk about 3 different strategies that we've been pursuing for engineering scaffold-free tissue constructs: modular assembly, cell-only bioprinting, and 4D strategies. So as this audience knows, the traditional strategy to engineer tissues has been very promising. So it typically involves combining, I'm going to change this to a laser pointer. Typically involves seeding cells and or bioactive factors within a biomaterial scaffold, and then this construct can be implanted into the patient with signals from the scaffold and the bioactive factors and the defect itself regulating cell behavior to promote tissue repair. Alternatively, this construct can be cultured in vitro in a controlled bioreactor system with regulated temperature, pH, perhaps application of mechanical stimulation or electrical stimulation to try to grow the tissue outside of the body, and then this more mature tissue can then be implanted into the defect. Now, the strategy is very promising because the properties of the biomaterial can be tailored for specific applications. One can control the physical properties of the scaffold, such as its swelling, degradation, mechanical properties. One can control the biochemical signals presented from the scaffold, such as cell adhesivity. One can deliver a wide range of different bioactive factors, such as growth factors or genetic material, and all these signals can ultimately be used to regulate cell behavior to enhance the rate and quality of new tissue formation. But there are some potential drawbacks with this approach. Ideally, with a scaffold-based strategy, one wants it to go away in concert with new tissue formation, but it's often very difficult to synchronize scaffold degradation with new cell division and new extracellular matrix elaboration. In addition, the biomaterial or its degradation products might present potential toxicity or immunogenicity, and importantly, the biomaterial may interfere with cell-cell interactions that have been shown to be critically important during development and healing. And it's for these reasons that a large portion of my lab has been focusing on cell condensation or high cell density system tissue engineering. Now, cells have transmembrane receptors called cadherins that are on the surface of cells. And when cells come in physical contact with neighboring cells, they form a physical bond. If one takes a suspension of cells and lets them settle due to gravity or centrifuge them down in a multi-well plate or a tube, when the cells come in contact with each other, they physically connect with each other and form a self-containing mass or aggregate of cells, typically spherical in geometry because this has the lowest free energy, but a wide variety of different geometries can be formed, such as sheets or rings or tubes, and even more complex architectures that I'll talk about later in my seminar. Now, the strategy avoids some of the negative microenvironmental signals that can be present when using scaffold-based approaches. Importantly, it's a more biomimetic environment because there is not a biomaterial interfering with the formation of abundant cell-cell interactions. And again, since there's not an interfering biomaterial or scaffold, the cells are free to degrade, remodel, secrete extracellular matrix at whim. And so the cell source that I'm going to be that most of the work that I'm presenting today involved are the clinically relevant mesenchymal stem cells. So these cells can be obtained from a wide variety of different sources, such as bone marrow, fat, muscle. We obtain ours predominantly from bone marrow. They can be expanded extensively without losing their multipotentiality, but when given specific signals, they can be guided to differentiate down numerous different connective tissue lineages, such as the osteoblastic lineage for bone, chondrocytes for cartilage, myoblasts for muscle. So our lab has pursued this high cell density strategy for engineering a wide variety of different tissues, and I'm going to present some of our work on engineering cartilage and bone, trachea, and vascular tissue today. So there are over 2 million bone graft procedures performed each year throughout the world, and millions of people suffer from major bone fractures, and about a quarter of these are going to have problematic healing because there is impaired intrinsic repair. On top of this, millions of dental, oral, and craniofacial procedures are performed each year throughout the world. And there are major issues here. For example, 86% of adults over 70 years old suffer from periodontitis, and this is loss of the underlying bone and soft tissue that holds our teeth in place. And this ultimately contributes to about a quarter of adults over 70 years old losing their teeth. So there's a great need for bone tissue engineering strategies. Now, we wanted to take some clues from how nature forms bone. And bone is formed naturally during healing and developmental processes, during healing development through intramembranous ossification and endochondral ossification. Now, in intramembranous ossification, the mesenchymal stem cells directly differentiate into osteoblasts and form bone. And it may be challenging to use this type of process in a tissue engineering strategy because osteoblasts have high metabolic needs. They need a lot of oxygen and nutrients. And initially, there's not a vascular supply in a typical engineered construct. So if one engineers a large engineered construct, the cells on the interior will lack oxygen and nutrients and likely apoptose. In the endochondral ossification process, mesenchymal stem cells first differentiate into chondrocytes and form a cartilaginous template, or called an anlaga. And then over time, this cartilaginous template is remodeled into new bone. Now, this strategy may be advantageous for engineering bone because one may be able to engineer a larger piece of a larger construct, with the initial chondrocytes forming having lower metabolic needs than osteoblasts, and so those cells on the interior can survive long enough to allow for vascular infiltration and remodeling of this cartilage template into bone. Now, there have been several groups that have demonstrated that condensations of mesenchymal stem cells can be driven to form new bone tissue through the endochondral ossification pathway. These strategies typically involve culturing in a chondrogenic media for 2 to 4 weeks, and then transitioning to a hypertrophic or an osteogenic media for an additional 1 to 3 weeks. And this typically results in a cartilaginous core and a thin bony shell surrounding that cartilaginous core. But importantly, in this system, the instructive signals guiding cell differentiation are presented external to the cell condensation. And so the cells on the interior have a lower concentration of these factors due to diffusion limitations, and the cells on the exterior are constantly taking up growth factor, and that further depletes the concentration that may be available to the cells on the interior. And on top of that, these constructs have to be cultured extensively in vitro prior to in vivo implantation, and that would delay a clinical strategy. And so we wanted to see if we can incorporate some of these instructive cues within the cell condensations themselves. And so here we turn to delivering potent chondrogenic factor TGF-beta1 within gelatin microspheres, and we wanted to deliver this factor relatively quickly to induce formation of a cartilaginous template. And then we wanted to deliver potent osteogenic factor BMP2 using mineral-coated hydroxyapatite microparticles, and we wanted to present this BMP2 over a more extended period of time to facilitate the remodeling of the cartilage template into bone. So gelatin it can be made into a biocompatible and biodegradable microgels using a simple emulsion process. And the acidic gelatin forms ionic complexes with the positively charged TGF-beta1. In this system, importantly, we can achieve 100% incorporation efficiency of the growth factor into the microparticles and of the microparticles into the cell condensations. And the growth factor release is cell-mediated because the cells release collagenases that ultimately break down the gelatin, and that then releases the TGF-beta1 that it is affinity bound to the gelatin. And we can control the release rate by controlling the extent of cross-linking with natural cross-linker genopin. And this is the morphology of the gelatin microspheres, typically spherical in morphology, with low and high cross-link microspheres having an average diameter of 65 and 53 microns. For delivery of the BMP2, we teamed up with Professor Bill Murphy, a good friend and collaborator from the University of Wisconsin, and he's established a wide range of very novel biomaterials that have a mineral coating. So his group incubates biomaterials in a simulated body fluid to form a mineral coating on the surface of the biomaterials. And this mineral coating has an affinity for a wide range of different bioactive factors. And by regulating the content of this coating, one can control the release rate of these factors. And in this example, by regulating the carbonate concentration in the mineral coating on hydroxyapatite particles, we can control the release of BMP2 over the course of 4 weeks. Now, since it had not previously been reported that exposure of mesenchymal stem cell aggregates to TGF-beta1 chondrogenic media and BMP2-containing osteogenic media could induce bone-like formation through endochondral ossification, first we presented these signals exogenously to the cell condensations. So we cultured first in TGF-beta1 containing media and showed strong staining for a GAG using SAF-O, no mineral staining with alizarin red. And then we transitioned to an osteogenic media containing BMP2 and obtained even stronger staining for GAG, along with bone-like mineral formation. And we have biochemical data supporting these histologic findings. We then removed the growth factor from the media and incorporated the TGF-beta1 releasing gelatin microspheres and BMP2 containing mineral-coated hydroxyapatite particles. And we were able to show robust GAG formation by 2 weeks, some mineral staining at 2 weeks, but that's just because the hydroxyapatite particles contain calcium. And then by 5 weeks, we saw a reduction of the GAG and a substantial increase in the bone-like mineral formation. And interestingly, this bone-like mineral was forming in regions where we were seeing reduced GAG, providing some evidence that the GAG was potentially being remodeled to form this new neobone-like tissue. And again, we have biochemical data that supports these results. So we then wanted to see, could we use this strategy to try to repair bone in vivo? And there have been several groups that have used mesenchymal stem cell condensations driven to undergo bone through endochondral ossification. But in all of these systems, they required 3 weeks or longer of in vitro culture prior to implantation. And we wanted to ask the question, if we incorporated these instructive signals within the condensations themselves, could we deliver these constructs at a much earlier time point, at just 2 days? So to try to answer this question, instead of engineering aggregates, we then tried to engineer bone-like sheets that would be more clinically relevant and more easily applicable to calvarial defects. So we formed sheets and assayed them at 5 weeks and showed strong markers for chondrogenesis with GAG staining and type 2 collagen staining, along with strong osteogenic markers of mineral, type 1 collagen, and osteopontin. And then we took these sheets and we implanted them into full thickness, critical sized calvarial defects in athymic rats. And importantly, we implanted them after just 2 days of culture. And then we assayed the defects at 4 and 8 weeks using microcomputed tomography and histology. And here I'm just going to show the microcomputed tomography results, where we see that the empty sham defect had some bony apposition around the periphery at 4 and 8 weeks, but really overall minimal bone formation. In contrast, at 4 weeks, in the defects receiving stem cell sheets with both growth factors, we observed complete fill of the defects at 4 weeks with continued bony apposition at 8 weeks, with significantly greater bone volume to total volume within these animals compared to sham empty defects, and also compared to defects that received sheets that only had TGF-beta1 and not the BMP2 delivered within them. So then with that initial success, we wanted to see if we could answer a more basic biologic question. So it's been known for over 100 years that mechanical forces influence both bone formation and bone healing, but very little is known about how the in vivo mechanical environment of a bone defect influences the capacity of transplanted cells to heal that defect. And so to try to answer this question, we teamed up with Professor Joel Burkell back when he was at the University of Notre Dame, and now he's at the University of Pennsylvania. And so he's developed some very elegant fixators that allow for regulation or control of the in vivo mechanical environment within a bone defect. So in this study, we formed a femoral defect in the rats, so an 8 mm femoral defect, and then applied a typical stiff fixator that shields the defect from loading during normal animal ambulation. We also had a condition using Professor Joel Burkell's unique fixators that have an elastomer here that allows for imparting mechanical forces within that defect immediately after surgery. And we call this the early loading group. We can also apply a rigid clip to this early loading group to stabilize the defect again, and then remove the rigid clip at a later time point, in the case of this study, after 4 weeks, to allow for delayed loading of the defect. And so in this study, we implanted the mesenchymal stem cell sheets, receiving only the gelatin-loaded TGF-beta1 microspheres to try to kickstart endochondral ossification for the formation of cartilage. And then we want to see how the in vivo mechanical environment influenced the healing of these bone defects. And so we were able to demonstrate that there was significantly more bone formed in both the early and delayed loading groups at 8 and 12 weeks compared to the stiff fixator group. In fact, in the stiff fixator group, by 12 weeks, there was not complete bridging in any of the 8 legs studied in this group. In the early loading group, we had complete bridging in 2 of the 8 defects, whereas in the delayed loading group, we had complete bridging in 6 of the groups. And there looks like there's a question in the chat, so I'm going to open that real quick. See if that's. Okay. Thank you for the question, Rahime. How do we make the sheets of cells? How do you control the shape and thickness of the sheet? Did it have even thickness? So we made these sheets by seeding the cells and the microparticles on a transwell membrane. The sheet, it was a 2-dimensional planar, and the thickness was relatively uniform. We can control the thickness of the sheet by regulating the amount of cells and the amount of particles that we use to form the sheets. And how do we control the diffusion problem? So within this system, there was not a diffusion problem because within the transwell membrane, there was media both below and above the formed sheets. And the sheets that we formed were approximately a millimeter thick. And since they were initially undergoing chondrogenesis, they had lower metabolic needs, and we didn't see any cell death within these constructs. So thank you very much for those questions. So we wanted to also examine the functional outcome within this study, and so we performed torsion tests on these bones, and we quantified that there was significantly greater torsional stiffness and failure torque in the delayed loading group compared to the stiff group. And in fact, there was not a significant difference in either of these variables compared to unoperated controls shown here in gray, even though there was quite a bit of standard deviation within the samples. Now, it may even be desirable to try to use this strategy to deliver genetic material instead of growth factors. So the growth factors, as you know, are temperature sensitive and also prone to degradation by cell-secreted enzymes. And so we wanted to see if we could deliver DNA encoding for the TGF-beta1 and BMP2 and incorporate these particles within the condensations. We wanted to see whether we couldn't turn the mesenchymal stem cells into machines producing these 2 growth factors of interest and driving their own endochondral ossification. And so here, we absorbed the plasma DNA encoding for both of these growth factors under those same mineral-coated hydroxyapatite particles and incorporated them into the mesenchymal stem cell condensations. And we were able to show that when we incorporated both the plasmid TGF-beta1 and BMP2 within the condensations, we could achieve TGF-beta1 production by the cells over 16 days. And similarly, when we incorporated plasmid for BMP2 onto the particles and into the aggregates, we could achieve sustained presentation or production of BMP2 over 16 days. And while I'm not showing the biochemical data, I'm showing the histologic data where we observed significantly greater bone-like tissue formation in the constructs receiving either the BMP2 plasmid or both plasmids together. More questions? So before I get into the next section of the talk, I want to address that question, although it's giving me a hard time. There we go. How did you control the release of TGF-beta1 and BMP2? So a great question, thank you. So within the system for the gelatin particles, the TGF-beta1 is affinity bound to the gelatin. And so we regulated release by the degradation of the gelatin, and the gelatin is degraded by the cell-secreted collagenases. And the rate of degradation is a function of the extent of genopin cross-linking that we use. So we use the natural cross-linker genopin. And so more highly cross-linked gelatin microparticles degraded more slowly. And so that delayed the release and presentation of the TGF-beta1 to the cells. And alternatively, for the BMP2 on the mineral-coated hydroxyapatite particles, by changing the composition of the mineral coating, we could change the BMP2 release profile and rate. Are they released sequentially or concomitantly? So another great question. So certainly not exactly sequentially. All that could potentially be ideal, and we're not sure. Within this system, it's not possible to not release them concomitantly, but we were able to achieve a more rapid release of the TGF-beta1 and a much more sustained release of the BMP2. Within this system, the TGF-beta1 releasing gelatin microparticles are completely degraded between 2 and 3 weeks, and the BMP2 hydroxyapatite particles still persist over the course of 4 weeks. And when we look at release in media of the BMP2, we can release for well over 4 weeks. So thank you guys very much for those questions. Okay, so now I'm going to transition to how we might use a Lego-like building block approach with condensations to try to engineer complex tissues made up of multiple different tissue types. And the example tissue that I want to present today is for the trachea. So we need an open trachea in order to breathe, but the trachea can stenose or close off for a wide variety of different reasons, such as the presence of a tumor, prolonged intubation, or tracheostomy, or external trauma. There are several different surgical strategies that can address this problem, such as removal of excess tissue using a laser or insertion of a tracheobronchial airway stent. But all these different procedures, ultimately, they typically result in restenosis within about a year. The only long-term strategy that has a successful outcome is tracheal resection and reconstruction, where the damaged part of the trachea is removed, and then the 2 healthy ends are anastomosed. And so this is a long-term successful strategy, but it's only applicable in adults if less than 50% of the trachea is damaged, and it's only applicable in children if less than 30% of the trachea is damaged. And so there are thousands of people worldwide that don't have a good surgical option. And so we turned to tissue engineering, and we had several main criteria that we ultimately want to satisfy. We certainly need the trachea to support an open airway. It needs to permit vascularization to bring oxygen and nutrients to epithelial cells that are lining the lumen of the trachea. And these epithelial cells will provide an immunoprotective barrier. And ultimately, we need that trachea to integrate with the recipient tissues. So to try to address this problem, we teamed up with, again, good friend and collaborator, Dr. Marcia Raleigh, back when she was at Worcester Polytechnic Institute, and now she's at Northeastern. So you can see a common theme here. We love to collaborate. And so if you do see areas where we might be able to team up, please do reach out. For this system, Professor Raleigh designed these cool agarose wells. So these are typical circular wells. However, they have a post in the center. So then when we seed mesenchymal stem cells and these TGF-beta1 loaded microspheres in the wells, here's a side view and this is a top view. The cells self-assemble around the post with uniformly distributed gelatin microparticles throughout their bulk. And then at 2 days, we can remove these rings of tissue and string them on silicon tubes or glass tubes like beads on a necklace. And if these rings are in close proximity, they'll fuse together, with the ultimate goal being the TGF-beta1 driving chondrogenesis of these cells to form a cartilaginous tube. And here we see the cells seated in these agarose wells, and then we string these formed rings on the silicon tubes. And then these rings and tubes that received either exogenous TGF-beta1 or TGF-beta1 through the microspheres underwent robust chondrogenesis, as shown here with this positive SAPH-O staining and type 2 collagen staining. And here's a native rat trachea staining for GAG. Now, these tracheal rings and tubes that formed had mechanical properties that were similar to that or even more robust than a native rat trachea. So here we have a rat tracheal ring and engineered rings without microspheres or with TJ-beta-1 loaded microspheres. And here we inserted two metal posts in the lumen of a ring and pulled those posts apart to measure a maximal loaded failure. And we were able to demonstrate that there was a significantly greater loaded failure in both of the engineered rings compared to the rat tracheal ring condition, significantly greater than or equal ring ultimate tensile strength compared to the rat trachea. And then when we fuse these rings together to form tubes, so here we have a native rat trachea, and here we have our engineered tubes with exogenous growth factor and delivered growth factor. And we turn these engineered tubes on their side and compress the lumen by 80%, and we measured significantly greater than or equal load compared to the native rat trachea at 80% collapse. And we could continuously compress the trachea lumen and achieve 100% recoil similar to the native rat trachea. But a movie may be more demonstrative. So here we placed the native rat trachea and our engineered trachea groups on their side and used a pipetter to compress the lumen. And we can see 100% recoil. These constructs are very tough and elastic. And it's important to remember that there was not a cylindrical scaffolding here. There is no biomaterial remaining within these constructs, and the mechanical properties are due to the cells and the newly elaborated extracellular matrix. And we can engineer these tubes a wide variety of different sizes, such as 2 millimeters, which is the average diameter of a rat trachea, 6 millimeters, which is the average diameter of a rabbit trachea, and even 12 millimeters, which is the average diameter of a human trachea, here in comparison to a native rabbit trachea. But we're under no delusions that we totally solved the tracheal tissue engineering problem, because the trachea is actually made up of C-shaped cartilaginous rings separated by a vascularized connective tissue. And again, the lumen is epithelialized. And so we wanted to see if we could make prevascular aggregates or rings by co-culturing HUVECs and mesenchymal stem cells in a vasculogenic media. And as early as 8 days, we started seeing these prevascular networks or structures. So these prevascular networks likely don't have lumens, and they're certainly not being perfused. But the idea here is that these prevascular rings might aid in anastomosis of the constructs more rapidly with host vasculature. So we then fused chondrogenic rings with these prevascular rings and cultured in a mixed media, and we were able to maintain a chondrogenic phenotype within the cartilage rings with positive staining for GAG, and also these prevascular structures is stained with CD31 within the prevascular rings. We then implanted these constructs subcutaneously in mice, and after several weeks, we injected the mice with a green lectin stain that would only stain the human cells. And when we isolated these constructs and performed histology, we observed these green circular regions, indicative that, and these would only have formed if the prevascular structures formed with the human cells had anastomosed with the mouse vasculature. And we've also seeded human bronchial epithelial cells on the cartilaginous sheets and been able to maintain the dual phenotype with positive staining for SAPHO and type 2 collagen within the cartilage sheets and positive staining for cytokeratins with the human bronchial epithelial cells. It looks like there may be another question, so I'll go to the chat. Have you ever tried using gels to release avastin or other angiogenesis molecule to help tissue vascularization? So we have not used the gels to release that drug. We've delivered other growth factors and genetic material, but certainly would be interested in talking with you if you have application for any other biologics that you're interested in. So up till now, the geometries of these cell condensations that we've been pursuing have been relatively limited, so rings and tubes and sheets. And it may be valuable to have the capacity to engineer even more complex architectures, to engineer tissues that have more complex geometries. And to do that, we turned to 3D bioprinting. So in the earlier days of tissue engineering, 3D bioprinting had been used to print, for example, thermoplastic scaffolds at very high resolution and stable structures. But then cells were seated on these scaffolds, and this presented relatively a 2D environment to the cells. And so that's not similar to the native environment. Over the past 15 or so years, a wide range of exciting strategies have been put forth for incorporating cells into macromer solutions and then printing these solutions and polymerizing them so that we have the cells encapsulated within the hydrogels, presenting a 3D environment to the cells. But again, this hydrogel then prevents physical interaction with the cells, and it impedes these cell-cell interactions. And so that may not be ideal if we're wanting to try to recapitulate some aspects of development and healing. More recently, there have been some reports of pre-assembled cell aggregate printing, an example of which is the Kenzen method, where cell aggregates are preformed, and using a robotic printer, they're placed in defined positions on these shish kebab-like skewers in close proximity to each other so that they fuse together to form more complex macroscopic geometries. Advantages here are that these systems are truly scaffold-free, and they allow for abundant cell-cell interactions. However, the resolution is limited by the size of the aggregates that are initially made and printed, and these condensations, cell aggregate condensations, ultimately require a pre-culture time of typically 2 to 3 weeks, at least, prior to the print taking place. And so we want to see if we could develop a strategy that would allow for immediate printing of individual cell-only bioink. And so to do this, we developed a novel slurry bath based on a dual cross-linkable alginate that we developed in the lab. So alginate is a natural polysaccharide derived from seaweed, and it typically can be cross-linked ionically using divalent cations such as calcium. And so we use this alginate to form calcium cross-linked microgels that are the basis of this slurry bath that has shear thinning and self-healing capacity. That means that it behaves solid-like when printing isn't taking place, but when, for example, a cell-only bioink is being extruded, it behaves more liquid-like, and when the needle is done being extruding, that local region behaves more solid-like. Now, since it's dual cross-linkable and we've added methacrylate groups to this alginate, and that allows for these microgels to be cross-linked together under the presence of low-level UV light. So after the printing, we apply low-level UV light to further stabilize the slurry and the printed design to allow the cells that are in close proximity to each other to physically couple through their cadherin interactions and form cell condensations that are in the geometry of whatever the CAD design file presents. And so this is just data demonstrating the shear thinning and self-healing behaviors of the slurry bath. As we increase shear strain, the storage modulus drops below the loss modulus, and the slurry becomes more liquid-like. Similarly, as the shear strain is increased, the viscosity of this bath goes down. And if we increase the strain, the storage modulus drops below the loss modulus, and the slurry bath behaves liquid-like. And then if we release the strain, we have an immediate flip-flop, and then the slurry bath behaves more solid-like, and this process can be repeated infinitum. So we can print any geometry that we're interested in, a cell-only printed letter C, a cube, a femur, apply UV light to photocrosslink the slurry, and stabilize these cell-only prints. The filaments, we're not limited in what kind of filaments we print, from a straight line to a 90-degree angle to an X. We can maintain high cell viability as demonstrated by these live dead staining images. And the resolution is similar to the inner diameter of the needle used. Here we used a 27 gauge needle with an inner diameter of 210 microns, and the average diameter of the filaments is about 276 microns. We can then print our constructs and culture in a differentiation media. For example, we can print a miniature femur and culture in osteogenic media, and then either design the slurry to degrade over time hydrolytically or manually remove it. And after 4 weeks, we can assay these constructs. Here we show alizarin red staining, indicative of immature neobone formation. And these constructs are cell condensation formed tissues. There's not a residual biomaterial present here. We can also print a miniature ear and culture in chondrogenic media, and then harvest the cell condensation formed tissues at 4 weeks and stain for GAG using toluidine blue O. And so in this strategy, we can use cell-only bioprinting to print 3D structures using a shear thinning and self-healing support bath. And the support bath can be stabilized to allow for precise maintenance of the geometry presented by the CAD file, and ultimately allow for the maturation of the tissues. So in the last part of my talk, before I get there, let's go to the question and answer section. Let's see. So we've got, I have perhaps a naive two questions. Are these MSCs different? As these MSCs differentiate, aren't they becoming immunogenic? So there's some evidence that that the MSCs may be immunoprivileged or even immunomodulatory, but the idea here ultimately we'd like to use host or patient autogeneic MSCs, and in terms of directing the proper differentiation, that is a function of the signals that we're presenting to the cells. In many of the cases that I'm presenting here, it's a presentation of growth factor or genetic growth factors like TGF-beta, BMP2, VEGF, or genetic information like the plasmids. How do you make sure the bioprinted organ has similar tissue functions, organ function to truly organ tissue? So that's going to be tissue specific. And so for some of the tissues that I was mentioning there, the mechanics is very important. And so we're currently working on trying to improve and tailor the mechanical properties of these tissues. Currently, we're trying to look at the role of the print parameters and the slurry on the mechanics of the formed constructs. So can you please elaborate on the strength of these tissues? So I don't have the mechanical data in front of me, but they're rather robust and rather stable. I know that's unsatisfying, but if you contact me afterwards, I can present you on some of the mechanical data of the printed tissues. I'm assuming you're talking about the printed constructs. The support path was made of the dual cross-linkable alginate, and so that's the both ionically cross-linkable alginate and the light cross-linkable alginate. And I'd be happy to talk to you more about that slurry as well after the talk. How fast is the cross-linking happening? So for the calcium cross-linking, so that can happen very rapidly if you're talking about forming the initial microgels. And so we use calcium sulfate here, so it's not as fast as calcium chloride, but we can tune that cross-linking. And the photo cross-linking similarly happens relatively rapidly. I have to look back in the paper whether it's I think we cross-link the BAP for 1 minute, but it may be slightly longer. How do you avoid immune rejection response happening when the bioprinted organ is in the organ transplantation? So in a lot of these strategies, we're looking at them as autogeneic cell transplantation. So the mesenchymal stem cells can be obtained from the bone marrow. The endothelial progenitor cells may be obtained potentially from blood. And so we're looking for autologous transplantation to avoid the potential immune rejection response, or potentially have immune matching donor cells. Okay, so in the last part of my talk, I'd like to discuss some of our more recent work on 4D biosystems to induce geometric transformations in cell condensation tissues over time. So I'd mentioned that developing and healing tissues typically begin as cellular condensations, and it's the spatiotemporal changes both in tissue geometry and transformations in the spatial distribution of the cells and the extracellular matrix that are essential for the evolution into a functional tissue. Now, 4D materials, which for the case of this talk are 3D materials capable of geometric changes over time, may have the potential to partially recreate these biologic phenomena. However, most reported 4D materials to date are not degradable or not biocompatible, and so that limits their application in tissue engineering. And there have also been no systems allowing for the control of geometry over time of high cell density condensations and their subsequent differentiation. And so to try to tackle this, we first looked at a relatively simple bilayer system. So here we incorporated up to 100 million cells per mil in the dual cross-linkable alginate macromer, placed it between two quartz plates and applied UV light. And then we incorporated up to 100 million cells per mil in a gelma layer, so methacrylated gelatin, and then cross-linked that layer on top of the dual cross-linkable alginate layer. And then when we punched out a construct and incubated in aqueous media, the alginate layer swelled much more rapidly than the gelma layer. And because the two layers were fused together, that induced a shape change or curling in this case. And we can maintain a very high viability of cells in both of these layers and we can visually observe the bending process over the course of time, here over the course of 14 days, and quantify this bending that occurs. Importantly, we can tune and control the rate and extent of bending by regulating the macromer composition of the layers, the thickness of the layers, and the concentration of cells within the layers. But within the system, it's not a true cell condensation, right? Because we have, again, hydrogel that is in between the individual cells, preventing at least initial direct cell-cell physical interaction. And so we had to develop quite a few other systems to be able to actually engineer a 4D cell condensation system. But before I look and tell you about those systems, I just wanted to briefly share a slight extension on that previous system, where we added an additional layer in order to allow for multiple step shape changes that might allow for the mimicry of dynamic tissue morphogenetic processes. So here we use 3 layers, and in this example, the red layer swelled faster than the other 2 layers, and so that induced bending in the right direction. And then when the red layer degraded, we had recovery of the initial shape. Then the blue layer swelled faster than the green layer, inducing bending to the left, and then degradation of the blue layer to recover the original shape. And we can use this approach to engineer, for example, a gripper arm, where the gripper arm bends upward, recovers its original shape, and then bends downward and recovers its original shape. But we may not be in need right now of a whole bunch of gripper arms. And so by using this approach, we wanted to see whether we could partially replicate a developmental process such as branching morphogenesis that occurs in many organs like the salivary gland and the lung. Now, these branching processes are not instantaneous. One doesn't have a sheet of tissue that then ultimately goes, changes into a fractal array of alveolar buds, say, for in the lung. Instead, it's a temporal process. And so by carefully choosing the placement of the different macromers within these layers, we could partially recapitulate this process. The initial formation of a bud, and then at a later time point, formation of a cleft, and then bifurcation, and the formation of 2 new alveolar structures. Now, this is not on the size scale of actual lung budding morphogenesis, and it's not on the same time scale, but it's initial proof of principle that we might be able to use such a system to study developmental processes, or even see if we kickstart some of the geometric changes that are relevant to these organs that might enhance our ability to engineer these tissues. Now, in the previous systems, we needed multiple layers of hydrogel in order to induce shape changes. And we wanted to see if we could induce the shape change with a single layer. So to do that, we incorporated a UV absorber along with the photoinitiator in the macromer solution, placed that solution between quartz plates, applied UV light, and because we have this UV absorber, there was a higher cross-linking density on the side closest to the UV light, and the lower cross-linking side farthest from the light. And when we incubate these constructs in aqueous media, the lower cross-linked side swells more rapidly and more readily than the higher cross-linked side, and that induces a shape change. And we could induce these shape changes in a wide variety of different materials: multi-arm PEG, GELMA, and the dual cross-linkable alginate. And we can combine this strategy with other tissue engineering technologies such as photolithography, ion transfer, and 3D printing to achieve even more complex final 4D shape transformations. Now, in the previous systems, you may recognize that the initial shapes were all 2D planar shapes. And so that ultimately may limit the complexity of ultimate 4D shape transformations that we can achieve. So we wanted to see if we could start out with a more complex 3D geometry, whether that would allow for more complex shape transformations. So here we turned again to our dual cross-linkable alginate to form microflakes that could be jammed upon extrusion and maintain their shape without printing into a slurry bath. And additionally, we incorporated UV absorber along with photoinitiator within this microflake slurry. Then when we applied UV light to a sample shape here, a gripper arm with a pillar, we applied UV light from the bottom and also from the left side. And then when we incubate an aqueous solution, the gripper arms then bend downward towards where the direction that the UV light was applied, and the pillar bends to the left in the direction to which the UV light was applied, the other direction the UV light was applied. So then we were ready to combine these systems to try to achieve true 4D cell condensations. To do this, we formed a gradient hydrogel layer that will serve as the actuation component of the system. We then printed microgels on a second layer and printed cell-only bioink within that microgel cell-supporting layer. We applied UV light to fuse the two layers together. And then when we incubated in differentiation media, the actuation layer could change the shape of the construct, while the microgel layer allows for cell-cell interactions and condensation formation. And the differentiation media could help mature the tissue so that ultimately when the microgels degraded, that would release the actuation layer and also release a more mature deformed tissue construct. And so we can print a wide variety of different geometries, such as a sheet, disc, bar, bar grid, net. And this just shows shape transformation of the cell-laden and cell-free constructs. And if we then make constructs that are cultured in chondrogenic media, but the experimental condition has an actuation layer, and the control condition has a bottom layer that doesn't have the UV absorber, so that bottom layer does not act as an actuating layer. When we culture over the course of 21 days, we achieve a shape change in the shape of a letter C here, only in the condition that had the actuation layer, but both of the conditions underwent chondrogenesis, as shown by mechanical testing, gag measurement, and gag staining. And so I hope I've been able to convey in my talk today some of our work that shows that inclusion of instructive signals in these high cell density condensations can be used to drive the formation of a variety of different tissues like bone, cartilage, vascular tissue. We can regulate the rate and extent of tissue formation by controlling how we're presenting the signals within the condensations. We can make many different clinically relevant shapes and sizes, such as a sheet, a ring, a tube, and even more complex architectures. This strategy may eliminate the need for exogenous bioactive factor delivery, which would allow for in vivo implantation more rapidly, which would save time and money and effort. And ultimately, these tissue engineering strategies can be used to partially recapitulate developmental microenvironments and processes such as branching morphogenesis or endochondral ossification. So they might be used to study fundamental mechanisms of development in addition to their use in regenerative medicine. And so with that, I'm very grateful to my group, especially those highlighted here in yellow who performed all the experiments resulting in the data I was able to present today. I'd like to thank my collaborators on the work that I presented today and these funding sources. And I'm thankful for your attention and these questions. I'd be happy to take any additional questions. We're always interested in hearing from potential new researchers that are wishing to join the lab and also new collaborations. So please don't hesitate to reach out.
Thank you so much for the fantastic talk, Professor Alsberg. Appreciate it. Couple questions people ask usually, what is the translational aspect? Are you ready to start clinical trials with some of these technologies, or how far are they off?
So great question. So the technologies that are at various levels, we're not ready to perform clinical trials using these strategies, but we do have strong data in small animal models, and we're looking to advance some of these technologies into larger animal models for ultimate clinical implementation.
Thank you. The other question is, when people deliver stem cells, usually, unless you have a delivery vehicle, they do not attach to the intended site and they die. What are your ideas? Do you prefer droplets, cell sheets, or other technologies to remedy this problem?
I'm a little confused by the question because I'm not familiar with
For cell delivery, when you talk about repairs, I think you mentioned cell sheets for bone delivering cells. What technologies, in addition to the you demonstrated, that people can use to deliver stem cells? Or it could be bone repair, or it could be other.
The initial part of the question seemed as to indicate that the cell that they've seen evidence that cell condensations cannot integrate without some sort of biomaterial carrier. Is that what I can't see the question?
Yes, yes.
Yes, I'm not familiar with that. So we do see, for example, in the bone repair that I demonstrated, where we can implant these cell only sheets, and they integrate with the host bone and form a continuous bony repair in the calvaria and in the femoral defects. And so I don't think that a biomaterial-based strategy of delivery is required for treating at least some tissues.
Thank you. Thank you. Regarding transplantation, sometimes some of the biomaterials may also incur transplant rejection. Do you have any thoughts on that?
Yeah, so that's a great question. So one has to be very careful in one's selection of biomaterials used. In our use with alginate, so we have not seen any rejection or immune response in the in vivo applications that we do the alginate, and similarly for the gelatin. And so yeah, so one has to be careful in the biomaterials that one is selecting.
So that's all the questions I have. Thank you again for your nice presentation, Professor Alsberg.
No, my pleasure. Thank you all for your attention, and thank you very much for your questions. I really appreciate it.
Thank you. Have a nice.
Thanks, Mehmet, and please take care. Say hi to Ali for me.
I will.
Thank you.